Hello everyone. So today we're going to demonstrate, or I am going to demonstrate, with regard to activity number 3, which is all about 1.4 flowers help plants to reproduce. Okay, so the aim of our investigation is to identify the different parts of a flower. Now, what are the things that we need? The first is we need a flower, of course. So the flower that we are going to use is a lily. Okay, so this is a lily. And then we're going to use a sharp knife. So here I have a blade. Okay, this is a blade of a cutter. Now always remember, when you are using this one, okay, any sharp material or object, okay, you should be use it with care, and there should be the supervision of an adult or of your parents. Okay, and then we are going to use a magnifying lens, okay, or also known as the magnifying glass. So as you can see here, those are the different parts of the flower that we studied in the class, right? And the flower smells so good. The scent is so sweet. So you know that they have this kind of scent to attract insects, right? Okay, anyway, so what we are going to do with this flower. So let's go to the procedure. In the procedure, you are being asked to remove and count the sepals, the petals, and the stamens from the flower. So unfortunately, we are going to separate the parts of this flower for the sake of experiment. Okay, so what are we going to do here? Now, it's not easy to identify the sepals of this flower. Okay, so I have here the flower bud of the lily. So as you can see, the color of the bud is yellow. So the one that surround the bud, they are the sepals, not this one. This one is a leaf. Okay, so they are surrounded with the sepals. So as we discussed, the sepals protect the flowers when it is a bud or still young and unopened. Okay, so therefore, if the flower already open, so this one already opened, so where are the sepals? Now the sepals will be the first 3, which are at the bottom of the flower. So that means the first 3 at the bottom will be the sepals. Okay, so there is no much difference on the sepals and petals of this flower. So the sepals are adapted to look like petals in order for them to have this, a lot of additional thing that will attract the insects. Okay, now let's remove the sepals. So we are going to remove the first 3. So those are at the lower. Okay, they are at the bottom of the flower. So we have the first one. Okay, so this is the sepal. And then the next one, which is at the bottom also, that's 2, and last one here, and that's 3. Okay, so we have 3 sepals. So the flower has 3 sepals. Then next one, let's go to the petals. So we have 1, 2, and 3. So we have also 3 petals. So if we are going to compare, so let's have a comparison. If we are going to compare the sepals and the petals of this flower, the petals are much larger. So they are much larger compared to the sepals, but they have the same color. So if you are going to draw the petal and the flower, just make the petal, okay, larger compared to the sepal. Okay, it is also wider on this part. Okay, so again we have 3 sepals. So 1, 2, 3, and we have 3 petals. Okay, now next one, we're going to remove the stamens. So the stamens, they are surrounding the part which is at the middle. Okay, so let's remove all the stamens of this flower. So we have the first one. Okay, 2, 3, 4, 5, and 6. Okay, so all in all we have 6 stamens, and as you look on one of the stamen here, so you have the anther, the one which is around what is this color, red orange, and then the one that supports the anther is the filament. So let's look closely on the anther using a magnifying lens. Okay, so you can see from there, that's not clear. Wait. Okay. Oh, the camera is adjusting. So you see that the pollen are ready to they are ready to, you know, do the transfer, the pollination. So they are going out already. They are going out already in the anther. So ready to, the pollen are ready to attach themselves to the insect once the insects will go to this flower. Okay, so that is the anther, which has the pollen, and the pollen is the male cell of the flower. Now next one, so we have 6 stamens. So let's just set those things aside first. Then the remaining part will be the carpel. So this is the carpel now. So in the carpel, of course, we have the stigma, which is at the top, and you can see there, okay, it's kind of there are some sort of liquid which is in the stigma. So that liquid, you know, it's going to it makes the stigma sticky. Okay, and then we have the tube that connects the stigma to the other part, which is at the bottom of the carpel. Okay, so we have the style here, and this is the ovary. Okay, this is the ovary of the lily flower. So the ovary is at the base of the carpel, and it is connected to the stigma with the style. Okay, so as you can see again, it's kind of, you know, it has some liquid coated on the stigma, so this will make the stigma sticky for the pollen. Okay, so let's try. We have here the anther, and okay, so let's try to stick this one here. So as you can see, the pollen sticked on the stigma. So therefore, the main function of the stigma is to trap pollen. Okay, for the pollination. So you are going to draw now 1 sepal and 1 petal. So just to refresh, we have here the sepal. Okay, it's narrow compared to the petal, which is wider. Okay, so the sepal, it's the function is to protect the flower while it is still unopened, like this one. Okay, when it is still a bud, and then the petal, it will attract the insects. Okay, so it is white in color. Then next one, so after drawing the petal and the sepal, you will go now in drawing the stamen and the ovary. Okay, the stamen or the carpel, I mean. So this is the stamen. So all you have to do is to draw okay, something like here. Okay, just draw this one, the anther, and then it will be supported by the filament. So you have to label your stamen, okay? Label the anther and label the filament. And after that, you need to draw also the carpel. Label the three parts. We have the stigma, the style, and the ovary, okay? Don't forget to label the parts. So after the drawing, we are going to cut the pistil, which is the other name for carpel, in half lengthwise, making sure that your fingers are out of the way, of course. So if your fingers are on the way, so they will be cut also, so we don't want that to happen, okay? So I always a reminder, class, when you are working with a sharp object, so be careful, and as much as possible it should be with the supervision of your parents or any adult, okay, for us to avoid accident. First, we are going to detach the carpel from the flower stalk, okay, so that's it. And then we are going to cut it lengthwise. So that means from the here we have the ovary, so we're just going to cut it lengthwise. Okay, and then we go to the style. Let's get to style lengthwise. It's kind of hard to cut. No, it's hard to cut the style evenly because it's not that big. So we're just going to cut it half all the way to the stigma. Okay, yes, nicely done. Okay, now we have pistils cut into lengthwise I mean, and then use a magnifying glass to look at the inside of it. You should be able to see a long hollow tube. So let's see with our magnifying glass if there will be, so even without the magnifying glass you can see clearly that there is a hollow tube at the middle of the style. Okay, so there is the hollow tube. Okay, the camera is adjusting so it turns blurry. Okay, so there is a hollow tube there. It is where the pollen will, the tube of the pollen will go. And then what is it called? So what is the name? What is the name of that hollow tube? So you have to write that name. And then what do you see at the bottom of the pistil? So at the bottom of the pistil, what do we call this? What is the name of this part, which is at the bottom of the pistil? And the next one, what does it contain? So what does the pistil has? Okay, so the pistil contains what? Now let's see if we will be able to see the ovules inside, if they are there. But I don't think we can see. We weren't able to see. I think the what is this? The magnifying glass is kind of blurry. Let me just wipe the glass first. Okay. Now let's see if we can observe the ovules. Okay, not that much, but you will see there a dark green at the middle. So at the dark green at the middle is where you can find the ovules of this flower. Okay, they are too small that you will not be able to see it even with the use of magnifying lens. Okay, let me crack this one. Okay, see those things? Wait. So those are where you can find the ovules. Okay, so again, So after that, you are going to draw the whole flower, and then you have to label all the parts. Okay, so let's have some review of the things that we studied about our experiment today. So again, the flower has 3 sepals. Okay, it has 3 petals. The sepals attract the insects, while the petals and the sepals, they protect the flower when it is still a bud. Okay, and then after that we have 6 stamens. Okay, we have 6 stamens. The pollen are ready to do pollination. You can see that it's even sticked on the table. Okay, and then it has, of course, 1 carpel or pistil. At the bottom of the carpel or pistil, we have the ovary, and then we have the hollow tube, which is the style, and then we have the stigma, which is, it has some liquid coating in order to trap pollen easily. Okay, so for the pollen, that is very sticky. Okay, so that's it for our investigation with regards with activity number 3, flowers help plants to reproduce. So I hope this investigation will help you, okay, understand better with regards with the different parts of the flower. Okay, so now with the remaining time, you have to complete your lab manual from pages 7, 8 up to page number 9. Okay, so you have to show me your drawing skills. Okay, so that's it for today, and start completing your lab manual now. Thank you.